begin.
Okay, thank you. Welcome everybody. Thank you for coming. It's a pleasure to introduce today Professor Tomaso Poggio from MIT. Dr. Poggio is a man who needs no introduction, but I will do it anyway. He's the Eugene McDermott Professor in the Department of Brain and Cognitive Sciences at MIT. He's a member of the of the CSAIL, as they call it at MIT, Computer Science and Artificial Intelligence Lab, and the McGovern Institute at MIT. He's an honorary member of the Neuroscience Research Program and a member of the American Academy of Arts and Sciences. He's a founding fellow of AAAI. He's a founding member of the McGovern Institute for Brain Research, and he received several honors, among them being the Laurea Honoris Causa from the University of Pavia for the Volta Bicentennial, for those of you who have heard the Volta, and the 2003 Gabor Award. He's just tremendously well known in the area of vision in general, and where he's introduced, of course, many key ideas, including the regularization math framework for ill-posed problems in vision, which is, of course, the stepping stone to learning from data. He has contributed to the early development of the theory of learning, introducing the radial basis functions, supervised learning, in reproducing kernel Hilbert space methods, and stability is a necessary and sufficient condition for generalization. He's had a great influence in modeling and visual recognition of the visual cortex, and today he'll be talking about the magic of the ventral stream, and it's a pleasure to have you here, and the floor is yours.
Thank you very much, Ahmed, for this introduction. Great to be here. I'll speak today about vision and recognition, object recognition, as a computational problem, and also about vision as an integral part of natural science, so engineering and science. These are the people who have been working with me in the last 12 months or so on what I'm going to speak, plus a number of others that have contributed to this work in the past. So when I started to be interested in vision, I was a physicist, and I've been working on vision now for about 4 decades. I've been working between brains and computers. And the reason is that I think I was actually interested in vision because my primary interest was in understanding the problem of intelligence. And I think that vision is one of the keys to eventually unlock the secrets of intelligence, what it is, how to reproduce it in computers, and how does the brain produce intelligent behavior. Now, the man who made this point better than anybody else is David Marr, here in the picture, who wrote this book called Vision, which is mostly wrong in the details, but is a wonderful framework for a computational approach to vision as an example of intelligent behavior. So I think the problem of intelligence is the greatest problem in science, more than the problem of the origin of of life, or the problem of the origin of the universe, or the problem of the structure of matter, and so on. The reason is one can argue about which one is the most important. I would argue for this to be the most important, because if you can make any progress in this, in terms of, for instance, improving our own intelligence, or building machines that can help us thinking more efficiently, then we can solve all the other problems more easily. So that's, in my mind, has the highest priority. And of course, what has been happening in the recent 5 to 10 years is that the old dream of artificial intelligence, of making machines as intelligent as we are, is starting to see, I think I may have lost a video, but it's not so important. There are now a few examples of systems that are better than us in narrow domains of intelligence. For instance, Deep Blue is better than us at chess. Google is better than any librarian could ever be. Watson is better than human champions at Jeopardy. And this is the X-47B, which is going to land by itself on an aircraft carrier this year. And this is probably the most difficult task for a pilot to achieve. So the point is that I think the next 5 to 10 years will be golden years to develop intelligent applications. For instance, a Siri who really understands my accent. That's my my example. This will come, but let me show you a couple of examples of the same type, kind of narrow domain of intelligence in the domain of vision. When we started to do this face detection, this was some of the very first example of this. Now, you take your phone and it does it for you, finding faces. About the same time, we started to do, in a project in collaboration with Daimler-Benz, Mercedes, a system to detect people, pedestrians, using machine learning techniques. And this is, for instance, the system in the trunk of a Mercedes in Ulm, Germany, detecting pedestrians from a car. And as you see, there are mistakes. At the time, there was probably a mistake every few frames, which we thought as scientists was very good, but of course practically unusable. Now there is a company, it's not the only one, but it's probably the best one, called Mobileye in Jerusalem, started by Amnon Shashua, who is a professor at the Hebrew University. He was a student and postdoc with me. And they have a chipset that they sell to Volvo, BMW, Toyota, Ford, and so on. And you can already buy some of the cars. It will break the car if you come too close to another vehicle or to a pedestrian. Here is an advertisement from Volvo, the S60 that you can buy since a year or so. It has a radar and it has a vision system like the one we started working on years ago. And this is just to show that this is useful. Yeah, I guess you get the point. So probably Amnon tells me that they have a project with BMW, but probably other ones, in 2 or 3 years, 2016, I think, 3 years, there should be an autonomously driving BMW with systems like this. So there are all these amazing applications, but none of them is really intelligent in the sense of passing a Turing test for vision, which is really, you know, I would ask to you, look, tell me what you see. And then I can ask a lot of questions. You know, is Ahmed, in which row is he sitting? You can imagine, a lot of questions I can ask. And there is no such system, we don't know how to build a system that could answer all these kind of questions that we can answer so easily just by looking at things. So what we can do so far is kind of answering this question of what is there in the specific domain. And so I think it's an interesting bet is will computer science by itself get to develop machines that are, you know, like us, that we cannot pass a Turing test, we cannot say whether it's a machine or a person, or it will be a combination of neuroscience and computer science that will do that. And my bet is on this, partly because the Turing test is rigged. Turing test is really a definition of human intelligence. If you have a computer that is much more intelligent than we are, it will not pass the Turing test because we will be able to distinguish it from a person, right? So I'm partly joking, but the story is that right now I think that this combination of try to use all we know about intelligence, including the brain, what we know about the brain, may play a key role also in developing intelligent machines. So let me tell you briefly about this project in my group, which started about 10 years ago for building a model of the ventral stream of visual cortex. So just to give you some feeling for the numbers, these are, of course, neurons in cortex. There are about 10 to the 11 neurons in the human brain, 1,000 times or 10,000 times more synapses. This is probably the most important difference with respect to computers. In a computer, typically there are 3 to 4 wires getting in or out of every element, like a transistor or a logical unit. In the brain, each neuron receives about 1,000, 10,000 inputs. Vision is a big chunk of the human brain, probably close to 30%, even more in the monkey brain, which is the closest model we have to us. And the ventral stream, here is a monkey brain, the front, the eyes are here, and information from the eyes goes to the back of the head, where there is primary visual cortex, and then it goes towards the front through this area called visual area 2, visual area 4, infratemporal cortex, and then other areas that are doing things like language or decisions or so, motor control. This is the ventral stream. This is plays a key role in recognizing objects. What do we know about that? There are in the monkey close to 1 billion neurons here. You know, this is kind of the box diagram. This is V1. The size of the box is roughly the number of neurons, and the size of this connection is roughly the size of the connectivity. You know, you are not a neuroscientist, I guess, mostly. Who is a neuroscientist here? Anybody? Right. So, you know, we know a lot about the brain and details. We know also very little, partly because we cannot really, we don't know the whole detailed circuitry. And even the properties of neurons are usually just by sticking an electrode in the brain and randomly finding a neuron that responds to a visual stimulus. And then, you know, of course, when you are sampling a few hundreds at most of these neurons out of 1 billion, it's so Huber and Wiesel worked in the 60s, 70s in V1 and won the Nobel Prize for their work. They found out that the beginning of visual information processing in Cortex, you have cells that are a little bit like Gabor filters, the product of a Gaussian and the sinusoids. And they are also a bit like edge detectors, oriented. So you have various orientations in each position of the visual field. Small receptive fields. This could be like a few minutes of arc, half a degree. My thumb at this distance is about 2 degrees of visual angle, just to give you an idea. And so they found what they called simple cells that are, as I said, kind of a different orientation of edges. And then they found complex cells that are the same, only that they are more invariant to the exact position of this stimulus. So when you find the stimulus for a cell, the cell will start firing, will be electrically active. If you, for instance, you are a different position or orientation, it will not be activated. And this one is more invariant to position and size of the stimulus. Okay? As you go up to V2 and V4 and so on, you find, this is a cartoon, but you find that the stimulus that stimulates best neurons in those areas get more and more complicated. And up to the most anterior part of IT, where you find also cells, for instance, actually patches of cells that respond to full faces, sometimes neurons that seems to prefer one face versus another one. So there is a gradual increase in the size of the receptive field. As you go up, you can, if you find the stimulus to which the neuron respond, it's very likely that even if you shift the position of it by several degrees, it will still continue to respond. So you get more invariance, translation invariance and size invariance as you go higher up. About 10 years ago, actually more by now, we developed a model based on data of that type and many other ones, based on the connectivity we knew from anatomy and physiology, and is essentially a multi-layer networks feed-forward in which you have Gabor-like filters, oriented filters here, and then you have complex cells that pull together simple cells with the same tuning preference, like orientation, and then by this pooling get more invariance to position of the stimulus. And then so you have like OR gates and AND gates going up, and OR gates and AND gates. I don't want to go into the details because that's a series of filtering and pooling operations, and it's not the first one of this type. Huber and Wiesel, in their original work, proposed in words an idea like this of having simple features and then more and more complex ones. You can imagine that at this level, for instance, the kind of filters here are more like corners, combinations of these filters here. In our model, these are hardwired and these are learned in an unsupervised way, but a very simple way. But the point I want to make first of all, it's not the first one of this type. There are several other ones. Probably the first one is really Fukushima. Neoconitron was called the model. It's probably the most faithful to the data and the most complex one. The software has been available and downloaded for quite a lot. It works pretty well for what we wanted in terms of we wanted to have essentially a compressed description and prediction of findings about neurons in different parts of visual cortex. And don't read the list, but it does a pretty good job in terms of either using some of the data for setting some of the parameters or predicting other ones. Here is an example of, in a sense, a prediction. Sorry, let me try again. So this is called rapid categorization. This is not the way one does the experiment with humans, but you can present an image very briefly. So you cannot move the eyes, there is no time. And then you're asked, is there an animal or not, for instance, a binary response. And this kind of model does essentially as well as humans. It's essentially on the same set is 80% correct. For humans is about the same for the model. And images that are difficult for humans are also difficult for the model and vice versa. This does not prove the model is correct, but more surprisingly is that this kind of models, at least back 7 years ago, was as good as computer vision systems, although this was not the goal that we tried to reach. So it was kind of an accident. So it did pretty well on the various databases like the Caltech 101 or this database of objects in streets of Boston that we collected. And there is a similar model based on very similar to this one. We are using it, or actually biologists are using it to automatically classify behavior of a mouse in a cage. So this is phenotyping, automatic phenotyping of behavior. It does essentially as well as human technicians in classifying micro behaviors like eating or walking and so on. This is important for biologists because there are thousands of mutants of mice. The genetics is known very well, the genomics, the gene sequences, but the behavior is usually only assessed qualitatively and not in a non-reproducible way. This is a way to make it reproducible and automatic. Now, so point is they work pretty well, this kind of models. And in fact, recently, like a few months ago, there were a model with a similar architecture of essentially feed-forward convolutional network filters, similar to Jan LeCun networks, was developed in a group of Jeffrey Hinton and did extremely well on a serious vision competition, ImageNet, which has millions of images and thousands of classes. And he did 10% better than everybody else, so that's quite something. And a similar type of architecture, again, convolutional, does very well also as pre-processing for speech recognition. So the question is why? You know, what? There is an intuition, but you know, I'm a bit ashamed to say that for 10 years I did not quite understand why they worked as well as it did. Understand means, you know, you have a model, you can simulate, you can change parameters, but as soon as you have more than 3 or 4 or 5 parameters, you know, it's impossible to answer a question like what is the optimal number of layers or under which conditions, you know, this is the optimum or not. It's just, so we need a theory in the sense of theorems, of really a characterization. And we tried with Steve's mail in an attempt. It was formally nice, but it did not bring too much. Here is another attempt. This one we've been working on for the last 16 months or so. And it's unpublished. And it starts with an assumption. It starts with the assumption that the main computational goal, motivation of this kind of architecture, particularly the ventral stream, this sequence of visual areas, is to compute a representation of images which is invariant to geometric transformations. Okay? The motivation is this. This is just a motivation, it's not a proof. Suppose I train a classifier with one example of one dog and one example of one horse. And then I want to test it, how well it's going to do on new example. You can do this with a human baby, you know, how well it's going to do. And if you do this with these images, turns out that one example per class, 3, 6, performance is 50% chance. Now, here, suppose that the classifier, instead of being given the images I showed, is being given a representation, say images, that are rectified for geometric transformations. So here all the dogs and horses are presented from the side with standard illumination, standard distance. Then you train on one example of a dog and one example of a horse, and you're already around close to 70%. These are very dumb classifiers on pixels. So this is just a motivation to say maybe a lot of the difficulty in object recognition, apart from clutter, is due to geometric transformations. It's not so much the variability between, for instance, different types of dogs and horses. This accounts for the fact that there will never be 100%, but it's coming from the viewpoint variability. Okay, so let's try to follow up this assumption. The assumption that the goal of ventral stream is to compute a representation which is invariant to geometric transformations. And it turns out that we can answer this and many more questions. So let me, for instance, one question is that it came up as a side effect of the theory is that in V1, as I mentioned at the beginning, Huber and Wiesel found evidence for simple cells being kind of Gabor filters. And nobody knows why they should be Gabor-like filters. Turns out that the theory I'm describing predicts this and predicts also specific ratio between the sigma and the wavelength of the Gabor filters. Interestingly, physiologists have measured these parameters in 3 different species, the mouse, the cat, and the macaque. And very surprising for biology, they all seem to be consistent across species. So that's a kind of universal constant which seems to scream for a theoretical explanation. And our theory provides an answer to this. Whether it's correct or not, we'll see. But so let me start with the fact that when you think about how neurons could represent an image, instead of thinking about pixel, you can think more broadly about measurements on the image taken by neurons, so functionals. And again, a motivation is thinking about which functionals is that neurons can very easily do dot products. That's, by the way, also why graphic GPUs, graphic processing units, are very good at simulating this kind of networks and neurons, because they can do dot products very efficiently. So if you have a dot product between an input X, say an image, and a template, a vector T, you can imagine that what is going on in terms of neurons is that you have inputs, and this will be the vector X, and the T will be the vector of synaptic weights. So essentially, there is no effort, and then the sum over the various products is because of summation of postsynaptic potential in the cell body, the soma, the so-called soma of the neurons. So the point is just dot products are a very natural thing for neurons to compute. Okay. So if you would have just to represent an image, you can imagine that what neurons at the beginning of the visual pathway could do is to compute a vector of features, and the features would be dot products of the image with templates, which are like filters, like Gabor filters, for instance, or anything else. So these are numbers. And of course, you know, there is this result that is, in a sense, the basis of compressed sensing and so on, that says that even or random projections, so if your templates are random images, say in this case, you can represent a high-dimensional image. Say D could be 1 million, for instance, pixels, with a much smaller number usually of projections, so K templates, and have a quasi-isometry, so the distance, the similarity between your images, U and V, would be kept within epsilon in this embedding, and furthermore, this number of templates you need depends logarithmically not on the original dimension, which is high, but on the number of images you want to deal with or distinguish. Okay, but that's just a comment. So the main transformation I'm going to deal with are a fine transformation of images, which are defined, and a fine transformation of this image means that I'm rotating or translating and scaling and combination of all of this. Okay, so suppose I want to have a description of an image which is invariant to a group of transformation, like the affine group, or say translation group, or whatever. Then one object that you can consider is the so-called orbit, which is the set of images that are generated by acting on an original one with all the elements of the group. Okay? So for instance, all the translations of one image. Translation, for simplicity, let's assume that is on a torus, so its coordinates are periodically, like an image that translates in the sense of going around me, around the observer. Okay. Now, the orbits are interesting because clearly it's invariant because it contains all possible transformation of the original image, this object. Could be an infinite number, of course, but it's also unique in the sense that if two orbits intersect, they are the same. Because if they have one image in common, Of course, from there on will be the same. So, okay. So now it's intuitive and can be proved that there is a one-to-one correspondence between orbits and the probability distribution induced by the group. So you can imagine that I could generate all the, for instance, rotations of one image and then take an histogram of all these images. Histogram in the sense of P of x1, x2, x3, where these are the pixels, for instance. So the order does not matter in the set of elements in the orbits, and that's what is embedded in the probability. So the sequence does not. Okay, so now there is that's interesting. I can have this high-dimensional object, the probability distributions in all these variables, which is invariant and unique. But how do I deal with that? Even estimating it, since it could be in 1 million dimension, if I have 1 million pixels, is a problem. There is another problem, which is that ideally I would like to be invariant to transformation of the image of your face without needing to see all the transformations of it, which is what I have assumed so far. I would like to see it once and be able to be invariant. Okay, there is this kind of thing solves both problems at once. So there is this theorem that says that if you have two high-dimensional probability distribution, P and Q, and I choose a projection at random, so T is also an image, then the one-dimensional probability distribution of the projections essentially characterize these high-dimensional distributions, in the sense that if these two are different, the probability that the projections are the same is very small, is 0. And if they are the same, the probability that PT and QT are different is 0. Okay? Now, in practice, you need more than one projection, and we have some estimates of how many you could need. So this is why I mentioned Johnson-Lindenstrauss before, because it's a kind of, in spirit, not in the techniques I used to prove it, but in spirit, it's a kind of projection of orbits. The most interesting part is that what I'm doing is I'm looking at the one-dimensional probability distribution. These are numbers generated. This is one template, the K template, and G, the probability distribution is over G. G is the random variable, is the elements of the group. And so this is a one-dimensional probability distribution. Because of properties of the dot product and the group, I can, this is equal to gi dot tk is equal to I dot the inverse of g tk. So I can move the transformation to the template. This means that I don't need to see the image of your face in all possible translations if I have a template stored before, which I've seen in all its possible translations. So that means that I can learn, let me see, I can learn, learning meaning, for instance, when you are a baby, there is the face of your mother on your crib, and it's moving and translating and maybe looming. You store these images. This will be all the transformation of one template. And that's all you need to do in order to learn in the future to be invariant to the same transformation for any object that you will see. So that's that's this point. And so they become a set of these things is a quasi-invariant which is associated with the orbit of this image. And so the idea is that during development, for instance, the visual system stores for each template, for k different images, stores each one of those images and all its transformations. And this is used later to produce a signature for any new image, a vector of these things, which is invariant, and out of which you can compute invariant quantities, like, for instance, I could compute histograms out of these quantities, empirical ones. I could, for instance, instead of this, compute moments of that one-dimensional distribution. So and this is, turns out this is one of the models that people have used for complex cells. Is the combination, the pooling of simple cells, for instance, square, the so-called energy model. This would be the second moment of these one-dimensional distributions. This would be a template like a Gabor function of vertical orientation. So, okay, is that more or less clear? This is, this has a little bit to do with neurons, but is also applicable and works in just as a computer strategy. In order to get this invariant representation, you just have a set of templates of which you know the transformations, and you are using them to obtain a signature that you can make invariant by group averages here.
So have people used these moments experimentally?
Well, for instance, the model we have used, this model I described before, was using essentially the infinity norm here. And yes, for the first layer was using exactly this. Only that the learning was not done in this way. It was hardwired.
Right.
So for translation, that's easy to do. The interesting point is that we can do it also for transformations that are not affine transformations. For instance, suppose you want to learn invariance to recognize the face of a person, invariance to expression. You can choose your template to be another face and G to be a change of an expression. It's not a group, so you don't get exact invariance, but you get quasi-invariance using the same machinery. And so what I described so far is really one layer of this, or the first two layers of the architecture. Is like the first layer and the pooling, the group average. But you can imagine that you can also have a layer on top of this, looking not anymore at the image, but at the neural image produced at the second layer. And the reason why you probably want a hierarchy is kind of computational constraints, connectivity. But the most important ones are these two. One is that you need to, when you do recognition, you would like to have an invariant description not only for the full image, but also for parts of the image, because I'm more interested in recognizing some parts of it, like your face here, than the whole scene. And so, in hierarchical architectures, let me see, like these ones, where you have essentially small receptive fields, so invariance over small, for example, translations here. This would be like a complex cells in view 1. This we can get invariance for small parts, an invariant signature, a vector of feature which is invariant. But then repeating the same operation here, I can get a signature which is invariant over larger ranges. And so I can have invariant signature at various levels of size and invariance. So I have parts and whole with their associated ranges of invariance. Okay, we have several results in the case of hierarchical architectures, but let me skip this in the interest of time, and let me mention another set of results that has to do with Y-Gabor functions at the beginning of the ventral pathways. So the idea is that, for instance, during development, probably also afterwards, there is this storage unsupervised of random images and transformations. And however, in neural terms, the idea that you store images is a bit not very plausible. It's like storing frames. What is more plausible is that you have synapses, contacts between the input from the retina to V1, and then you have a non-line mechanism in which each frame will change the weights of the synapses. And it's known that there are so-called Hebb synapses in visual cortex, in which the weight of the synapse changes in proportion to the product of the input and the output of the neuron. Now, it turns out that Hebbian synapses are a non-line version of an algorithm to compute principal components. It's Hebb plus a normalization term that you need in order to for the synapses not to remain finite. So this is Hebbian rule, and so this leads to the question: suppose that during development, the neurons see natural images shifting, say translation, basically because the receptive fields are small relative to the spatial frequency content of natural images. Every kind of translation at the level of these small windows is translation. Even rotation looks for most neurons locally as a translation. But anyway, it does not matter very much. But this leads to this problem. If you have Hebbian rule computing principal components, and you look at image translating through a Gaussian window, G, what are the eigenfunctions to which this algorithm will converge? So it's an eigenvalue problem. Find the eigenvalues and eigenfunction that solve this equation where G is the Gaussian density of synapses, and T is the autocorrelation of a natural image. And it turns out that this equation if this did not have the Gaussian, this would be Fourier components. With a Gaussian, it turns out that you find Gabor-like solutions. So of the form a Gaussian time complex exponential. And it's not exact, it's not a Gabor, but it's very close. And you can, of course, solve this numerically. And so these are the 0 eigenfunctions, the n equal 1 eigenfunction, n equal 2. So these are like odd Gabor-like function, even function. These are experimentally measured. And if you look at these plots, this plots the ratio between the sigma of the Gaussian and the wavelength of the sinusoid, and in the x direction, in which there is, this would be, well, in this case x would be this one, I guess, but, and this is in the other direction. The theoretical prediction, essentially parameter free, are the red crosses, and the other ones are measurements in cells in this free species, red and green cross. So there is a pretty good agreement, which again seems quite surprising, because the model was not developed to predict this, right? The model is really about recognition and invariant recognition. It's always very easy to have models predicting one phenomenon, but if this comes as a side effect, that's more surprising. And we have made prediction also for V4. I will not explain this, but for higher visual areas, assuming that the same machinery repeats in higher visual areas. And of course, the real test is to see whether implementing the whole network will get us results that are better than everything else. And we still don't know that. Everything else in this case means, for instance, the fact that we should be able to reduce by a lot the number of supervised examples that you need to learn by essentially learning in an unsupervised way the geometric transformations. There are other predictions we can make related to, again, a bit surprising. Do you have 5 more minutes? Yep.
Yes.
I mentioned this because this is very recently quite interesting. So this is, again, this is the brain of a monkey. This is the back of the brain. This is the front. And Doris Tsao, who is at Caltech, and Vinek Freivald, who is at Rockefeller, have found in the last 3-4 years that there are groups of neurons, the patches of neurons, in which the neurons are sensitive to faces. So exclusively to faces. They claim exclusively, may not be so exclusive. And the more interesting patches seems to be this one, which is the most anterior, which contains neurons that are essentially sensitive to the specific face, but invariant to pose, to viewpoint. And these ones, which are instead view dependent. So you go from view dependence to view invariance. And this machinery is probably almost certainly involved in identification, recognizing specific faces. Okay. Now, there is, so in terms of our, our approach, our theory, this would be on top of layers that have given you invariance to position, and then you'd have units that are specific to different views. The idea would be that they would be during development and seen a specific phase, maybe again the face of your mother under different views and stored representation of those. And then you get view invariance by pulling together different views. So this would be like the simple cells and the complex cells. Now there is an interesting, and if you do this, you get much more invariance to pose in recognizing a specific phase. So this is like presenting one view of a face, say a frontal view, and then checking how well you recognize that face among other faces for different views. As you turn away from zero, your performance gets worse and worse. But if you do this thing of having a template of a face, run another face for different views, you can generalize much better for pose. Not exactly, because it's not a fine transformation anymore. But because faces transform in the similar way, they have a similar 3D structure, they are almost, you can borrow information from one to another. So there is this interesting puzzle. Well, I can tell it in two ways, but one is that suppose there is this Hebbian rule at work in this kind of machinery, then you'd expect the simple cell here to be the eigenvectors of each specific faces over these changes, these transformations. In the previous cases, this will just shifts over natural images. Here is rotation in the image generated by rotation in depth of a face. And it turns out that since the faces are bilaterally symmetric, the eigenvectors are even or odd. You can prove that. And so the prediction is that you should get cells that, when probed with another phase, have tuning that is even. So either a peak in the center, or two peaks, or four peaks is symmetric. And and surprisingly enough, Winrich and Doris find that there is this area between this phase patch between the view tuned and the view invariant called AL, where if a phase responds to a view, where a cell responds to a view, it responds also to the mirror symmetric one, which is otherwise very puzzling, why you should have cells with this mirror-like property. So, so let me finish. So I'm a physicist, so one thing that I find pretty magic in all of this is the fact that, like in physics, symmetry invariances are key. You can essentially write essentially all equations of classical physics and quantum mechanics, from basic conservation laws and invariances to choice of measurement units and space-time invariance and so on. So here it would be kind of fantastic if from invariance properties that Cortex will have to have with respect to transformations in the outside world, you could really predict some of the architecture and the basic properties of neurons. I think it's too nice to be true, but if it's not true for the brain, it could be anyway quite interesting in terms of machine applications, system that can be trained with a small number of labeled examples. You know, one of the main so machine learning techniques can do very well in applications like the Mobileye one, for instance, I described, but they had teams in India labeling millions of images of cars and pedestrians because they need a lot of training. And that's the lesson. Also, the successful application needs a lot of labeled examples. That's okay, but it would be much nicer not to have to do that. And so that's one way that we think this could work, is the fact of having representation that are invariant to some of the most common transformations. And same for speech. You know, like, I'm sure I say one word, the same word in different ways, and different people can say the same word in quite different ways, and you can in this way learn these transformations, and potentially, we are trying to do it, we don't know yet how well it will work. Thanks.
Any questions?
So, do you think that the brain actually performs all of these integrals and complex exponentials and
Actually,
all that stuff?
no, absolutely not. But there is no need to do that. So all the operations that you need to do, are the brain needs to do, are actually very simple.
So how do you think, for example, that the brain does these rotation invariant transformations?
It does not need to do it. You see, you collect. So let me see. The simple version, which is not exactly what, is that if you store the neural image produced by one object that is rotating, and storing means storing in synaptic weights, then what the brain needs to do to have an invariant representation for a new object is to get the new image going through those synapses. You get a dot product, and you have stored several of them. So you have a set of dot products, these activities in neurons, and then you take the sum of those. The neuron takes the sum, and that quantity is invariant to transformation of the new image. So you don't need to do any of, the element of the group, the transformation, the brain does not need to know. It's implicitly in the series of images stored. There is no, you just.
Any other questions?
Humans quite good at the generalization. Let's say we see a chair, we see a bottle. Then once we see one or few, but we can tell others, which is quite different from this particular one. We can still tell that that's a chair, that's a bottle. My question is that how can you call synapses capture such a variation?
Well, you have invariance, but you don't lose the
evidence show that we can do that.
You mean that we can keep, if I see a face from different viewpoints, I know which face, but I also know the viewpoint.
Well, for example, if we have a regular face, and also we can tell from even Picasso's picture there are face in it, which is quite difficult to tell.
Yeah.
So we need some representation of the face that we can generalize.
Well, but this is a way to generalize, right? For instance, if you have a representation which is invariant to a range of affine transformations, this means that you can deform a face and will still be recognized as such.
Well, the assumption that we have such a representation
Well,
but
it's-
sometimes we may not. Right? How can we find such a good representation?
I'm just saying the representation is a set of weights.
Okay.
We can, we can reproduce that in a machine. There are other things. You mentioned chairs. So what I've been saying so far has to do with visual appearance. So there are very different types of chairs, which have really a different shape. And so sometimes we say, okay, that's a chair because we recognize this as a tool for sitting. So the definition is really functional more than appearance. So this does not explain that kind of higher level, categorization.
But to pick up on Lee's point, though, I mean, for instance, he was talking about Picasso. That the cubism, of course, is going to violate your fundamental, basically with all these discontinuities
Yes.
and everything. It's basically everything goes out the window, right? Those transformations are not gonna cut it.
Probably
because
not.
they violate your,
Yeah.
your
Strictly, it's a question of, it's related to the whole versus parts story. So, the reason for hierarchy is that I want to be able to recognize an eye by itself. And I also want to recognize a face in which the eye is a part, right? And so I can still recognize an eye even if it's separate from the face, if it's clear enough. So, it depends. When there are all these broken, deconstructed objects, I can still recognize the parts. And then I can make up a story about it.
I have one question. The two people, I forget the name,
Yeah.
one from Caltech and the other
Right.
one, where they
Doris,
supposedly
yeah.
isolated
Yeah.
the face-sensitive parts of the brain. Have they, that there's this condition where
Yeah.
humans, for instance, what's they call it, facial agnosia or something like that,
Yes.
where they can't recognize faces.
Yeah.
Have they checked? Have they sort of cross-validated with that that kind of people?
No, I don't think they did the lesions in the monkey, but so there are discussions. So that those face patches in the monkeys were found by doing functional imaging first, and then putting electrodes later. Now, in humans, functional imaging reveals also different face patches. And there are discussions about which ones are equivalent to which ones in the monkey, because our brains are similar but not the same.
Yeah.
So it's not completely clear. But there is evidence that in some of the patients, one or more of these humans now, one or more of these face patches, which probably correspond to the monkeys, is either missing or not active.
right?
Yeah. There are also interesting studies about development of this area from when you are a baby to higher up, how they become larger or smaller, or what the size of those is in autistic patients, for instance, because they're supposed to be less able to recognize faces or facial expressions. And so. It's a complicated story.
Yeah,
It's not.
that was actually the next question about the fMRI. So fMRI has helped in a sense along that.
Yeah. fMRI by itself is, of course, a very coarse instrument because one voxel is, like 100,000 neurons or so at once. And furthermore, fMRI is almost a miracle it works because what it measures is blood flow and oxygenation in the blood. And it turns out that this is closely related to activity of neurons, partly because of this miracle that there is a lot of very small blood vessels in the brain, so that it's like having a heating system but very dense. So because you're measuring, it's like if I would measure what my computer does by measuring where the heat is, and that level of indirect. So, but anyway, yes, fMRI has been useful. Yeah.
Any other question? Yes, Sean.
I was wondering that for human to recognize the object, do we have the concept of this shift model first, or do we have the concept of transformation first? I'm just wondering, for example, if I can recognize this model here, no matter what's the rotation angle, maybe it's because I already know I already have a prior information about the shape of the bottle. In other words, for example, if we have a very complex object here, and maybe we are not familiar with that, like a circuit board we never seen previously, and we rotated the circuit board, is that also simple for human to recognize these two objects are still, these two viewpoints are still similar, still from the same point?
Let's see, what has been a debate for a long time in cognitive science is whether when we recognize object, what we do is object-centered or is image view-centered. So, in other words, you know, object-centered would be more like a 3D representation, a model that we can then rotate in our head and so on. It seems that in terms of immediate perception, you know, when there are certain things which are more like reading a map. I have a 3D object and I have to see whether it matches this one or not. And then it takes a person a few seconds, and that it looks like this is more like rotating a 3D model in your head. But if you have immediate recognition, which is most of what we do, you know, it's not solving a puzzle, it's the bottle, the Coke, you know, then it's view-based. So you essentially recognize if you have seen a view which is close to the one you are looking at now, even if you're given stereo information.
I'm just wondering if we do the transformation by using the cortex or just using some other part of the brain. We handle the transformation by visual system, or we handle the transformation by the other part of the brain. Like it's like, for example, for shift translation, it seems pretty simple for us to detect. We can recognize,
Yeah.
no matter where is the object. But consider if we rotated a pretty complex object.
Yeah.
If it takes some time, I just don't know if it's still the visual system or it's
Right.
because the
So,
other part.
okay, so, you know, what I described, even if it's correct, is only 1/3 perhaps of vision, in the sense that it's only the feed-forward pass. You know, it's information going one direction. Now, there are a lot of back projections, and they are probably because, you know, this may explain what goes on when we see something very briefly, the first glimpse. But then there must be something more like a verification process. You know, is that really a face? You know, and then either you move your eye and you inspect the image, or there is a mechanism that's called attention, which is like focusing, processing in some parts of the image. And, you know, the idea that we're exploring with Shimon Ullman is that what is called attention is more like running some simple programs or routines back on the image for answering specific question or disambiguating certain things. So, you know, I think some of what we do is this, and it takes longer. And yeah.
All right. If there are no more questions, let's thank our speaker again.